Hi, Michelle Glass here. We are continuing with our Lecture 2 Part 2 series, and our topic here are those lipids. So we've already talked about carbohydrates, and here is our video on lipids. Now, lipids are your fats. So this includes fat, which is a semi-solid at room temperature. This includes oils, which are liquids at room temperature, and this includes waxes, which are solids at room temperature. So obviously our body is making both fats and waxes. You have earwax production from your ceruminous glands, but we're not really producing oils, although we do eat oils. So of course they're going to be important in our diet that way. So let us get started looking at our structure to our lipids. Now, as we discuss lipids, there's not actually really a monomer for lipids. You might list the fatty acid as a molecule, as a monomer, but technically it doesn't have one. Lipid structure is a long hydrocarbon chain. And so here we're going to see our ratio of carbon and hydrogen is 1:2. Now there's also oxygen that's part of the structure of our lipids, so we'll see that as well. Because we have a very long hydrocarbon chain, our lipids are described as hydrophobic. So they are non-polar molecules, which means they don't mix with water. There are lots of different functions to lipids. One includes its ability to be used as a source of energy. So we did see carbohydrates having energy source as a primary function. We see that lipids are kind of like our secondary energy source. So carbohydrates are the quick, rapid, kind of preferred choice by our cells. So about 60% of the time, our cells are using carbohydrates to make energy. And so then the remaining 40% of the time, our cells are using lipids. So this is a little bit of a longer to access source of energy. In addition, our lipids are involved in communication. So we'll see that some of our hormones, these are our chemical messengers that travel through the bloodstream, can be made from lipids. We will also see them important in structure. So we see that there are lipids that are composing the cell membrane. Our body, remember, is made of cells. So if the cell membrane is important to the structure of the cell, then obviously, it's also important to the structure of the whole body. But then also you've studied adipose tissue, one of your connective tissues in the lab. You know that that is your fat storage tissue. So it also has important structural components to the body. In addition, the adipose tissue is providing shock absorption and padding, so we can also include protection as one of the jobs of our lipid molecules. And adipose tissue is also providing insulation to the body. So we can include that also as a job for our lipids. When we talk about our lipids as being sources of energy, what we're really talking about are our fatty acid molecules. So that's our energy source. So just like we said, cells are going to break down carbohydrates into glucose and then use glucose to make energy. If we're breaking down lipids, so we're going to break it down into fatty acids and then use fatty acids to make energy. So here is a kind of diagram here of your saturated fatty acid. Saturated fatty acids have all single bonds between the carbons, and it's called saturated because the rest of these carbons are completed with hydrogen. And then we will have a carboxyl group on the end. So that's how it's called an acid, fatty acid. And going back to saturated fats, these are typical in animals. These are your semi-solid fats. Your oils, like in plants and fishes, are unsaturated fatty acids. And unsaturated fatty acids have a double bond. They may have just one, like what's represented here, or they can have multiple double bonds. This is called unsaturated because there's going to be fewer hydrogen atoms in an unsaturated fatty acid. The unsaturated fatty acids, the double bonds, kind of make a bend or a curve in the molecule, and that's what makes it an oil instead of a fat. Now, as we mentioned, fatty acids are the source of energy, but we can store fatty acids as glyceride molecules. So glycerides are the actual molecules that are storing fatty acids. That is what you are finding in your adipose tissue. That's what we're talking about here. Remember, we've talked about adipose tissues providing shock absorption and padding, energy storage, insulation, and we can add nutrient storage here because obviously if we're storing fatty acids and they're a source of energy, that's great. We also will have some lipid-soluble vitamins that can be stored in our adipose tissue as well. Now notice here, a glyceride is composed of a glycerol and 1, 2, or 3 fatty acids. So if you have 1 glycerol and 1 fatty acid, it's called a monoglyceride. If you have a glycerol and then 2 fatty acids, it's called a diglyceride. And if you have a glycerol and 3 fatty acids, it's called a triglyceride. So very often I'll just talk about triglycerides because it's like the fully packaged glyceride. Notice that to build a glyceride, you're going to do a dehydration synthesis. And then in order to break down glycerides, in order to release fatty acids, you're going to do a hydrolysis reaction. Okay, so we've mentioned several of the jobs of our lipids, and we included communication as part of that. There's actually 2 different classes of communicating lipids. We have what are called eicosanoids, which are like a localized sort of signaling system. So that means one cell is releasing it kind of like to the neighborhood, just to the surrounding cells, rather than being transmitted through the bloodstream. An example of an eicosanoid is prostaglandins, and these are involved in our pain signal. So a cell that's been damaged might release prostaglandins, kind of signaling to that local area that some damage has occurred, and that's going to help to bring in white blood cells and so forth. Hormones are different because they travel through the bloodstream instead of just being released to that little neighborhood. And steroids are an example of our communicating or our lipid hormones. These include our sex hormones such as estrogen and progesterone in females and testosterone primarily in males. Cholesterol is another example of a steroid hormone, and we'll see cholesterol is going to be an important part of our cell membrane. The cell membrane is composed not only of the cholesterol molecule, but also phospholipids, and then also what we can call glycolipids. Now, the phospholipid is going to be probably the most important component of this cell membrane for us as we progress in our studies. We'll talk just about phospholipids in the future. The phospholipid is going to create what we can call a semi-permeable structure to our cell membrane. Semi-permeable means some stuff can cross and other stuff can't. Now, the way that the phospholipid molecule is special is it's going to have a hydrophilic head and then a fatty acid hydrophobic tail. So the hydrophilic head is the water-loving, is polar, and then you're going to have a non-polar lipid hydrophobic region. And so that's going to be really important to understand as we start talking about our cell membrane more in the future. Those cholesterol molecules are going to help to stabilize the cell membrane so that it's not too fluid when we're looking at just those phospholipids by themselves. Glycolipids are going to be part of what we're going to call our glycocalyx, or you can call it the extracellular membrane. It's going to be both carbohydrate and lipid components. They're going to help to anchor cells to other cells as tissues, and also be an important in helping to get things binding to that cell membrane. So we're going to be talking about those again more in the future. And that's it for now.